Well, we have finished up skeletal muscle, and so now we start to transition into our second type of muscle, which is smooth muscle. And smooth muscle is going to be found in the walls of most of our hollow organs, with the exception of the heart. We're actually going to find that the heart has its own type of muscle called cardiac muscle. We typically will find smooth muscle in two separate layers. One is called the longitudinal layer, and the other is called the circular layer. Now, when we look at the longitudinal layer, when it contracts, it's actually going to cause dilation and shortening of the muscle and the organ. And when you look at the circular layer, when it contracts, it causes constriction and elongation of the organ and the muscle. One kind of caveat here is that we only find a small amount of endomysium in smooth muscle, whereas in skeletal muscle, we typically find a lot of endomysium. We also have a process known as peristalsis. And with peristalsis, this is an alternating contraction and relaxation of smooth muscle layers. And what it does, it helps to mix and squeeze substances through the lumen of hollow organs. The lumen is typically what we call the hollow hole or the opening through these organs. Again, going back to the previous slide, when the longitudinal layer contracts, the organ is going to dilate and shorten. When the circular layer contracts, the organ constricts and elongates. So let's look at the microscopic anatomy of smooth muscle. One of the caveats with smooth muscle is that the fibers are no longer rod-like or elongated like they are in skeletal muscle. With smooth muscle, the fibers are more spindle-shaped, which means they're more thin and short compared to their skeletal muscle counterparts. When we look at connective tissue, smooth muscle is only going to have small amounts of endomysium. Unlike skeletal muscle, which has all three layers, smooth muscle does not contain perimysium or epimysium. With smooth muscle, the sarcoplastic reticulum is less developed than it is in skeletal muscle. If we remember with skeletal muscle, the SR essentially surrounds all of our myofibrils. Not quite the same with smooth muscle. In smooth muscle, we also see pouch-like infoldings called caveolae of the sarcolemma. And these are there to sequester our calcium. In smooth muscle, we're also not going to see any sarcomeres, we're not going to see myofibrils, and we're not going to see T-tubules. So this is kind of a look at the different type of smooth muscle here. So when we look at the smooth muscle and a relaxed smooth muscle cell, you'll notice that they're a little bit more elongated, they're a little flatter, they're more spindle-like. And then you can see when they contract, they actually kind of shrivel up a little bit. This is essentially what we see with smooth muscle and layers of smooth muscle. And we'll talk about the components here in the next couple slides. So if we look at the innervation of smooth muscle and how the nervous system interacts with it, we have what we call varicosities, which are these bulbous swellings of nerve fibers. And these nerve fibers store and release neurotransmitters. And these neurotransmitters are going to have essentially a dual effect. They're either going to be inhibitory or excitatory, which can either stimulate a contraction or inhibit a contraction. Our autonomic nerve fibers are going to innervate smooth muscle at diffuse joints, and they have very wide synaptic clefts in the general area of the smooth muscle cells. Now, when we talk about autonomic nerve fibers, understand that autonomic means that you don't have control over it, whereas voluntary means you do. So autonomic is essentially involuntary smooth muscle or involuntary tissue, whereas skeletal muscle is voluntary tissue. When we look at the myofilaments in smooth muscle, the ratios are significantly different when we look at thick to thin filaments. The ratio of thick to thin filaments in smooth muscle is about 1 to 13, and that is much lower than we find in skeletal muscle. The thick filaments are going to have heads along their entire length, whereas in skeletal muscle, if we recall, the myosin heads are absent essentially in that H-zone region. Also in smooth muscle, there is no troponin complex. And instead of troponin, we have what we call calmodulin. And that calmodulin acts as the binding site for calcium. So again, no calcium going to bind to troponin and affect tropomyosin. In smooth muscle, calcium is going to bind to the calmodulin, which causes a muscular contraction. The myofilaments are going to be more spirally arranged so that when the muscle contracts or the muscle cell contracts, it actually corkscrews or contracts in a corkscrew manner. We're going to see that we have what we call dense bodies. And these dense bodies are proteins that serve as attachment sites for non-contractile intermediate filaments. They also act as anchoring points for thin filaments to the sarcolemma at regular intervals. So let's look at the location of where we find a lot of our smooth muscle. So it's going to be found in a variety of organ systems, also with a variety of roles. So if we look at the cardiovascular system, we're going to find it in the blood vessels. And this is going to help regulate the blood pressure and blood flow by constricting and dilating those particular vessels. If we look at the respiratory system, we're going to find smooth muscle in our bronchioles, which helps to control airflow to the alveoli, which again, essentially looking at constriction and dilation of the bronchioles. In the intestines of the digestive system, this is where we go through what we know as digestive peristalsis, and it's going to mix and propel our material. So as the intestines basically constrict and dilate, it basically is moving food through the intestine and mixes it together as we go through the process of digestion. We're also going to see smooth muscle in the ureters of the urinary system, which basically helps to propel urine from the kidneys towards the bladder, and then also tries to expel it out of your urinary system. And then we also find it in the uterus of the female reproductive system, particularly as we go through birth and delivery. So these are the steps that we find in smooth muscle contraction. This again will be a great graphic for you to look at. It should be in your book. So as we go through the steps here, always come back, and this is a great graphic to be able to look at and reference as you go through the steps. So as we look at the steps of smooth muscle contraction, the first thing that happens is we have some sort of neurological stimulation, and that is going to cause an opening of our voltage-gated calcium channels. As calcium channels open, that allows calcium to flood into our smooth muscle cell. And that calcium is going to bind to and activate our calmodulin molecule. The activated calmodulin activates a myosin light chain kinase enzyme. And this particular enzyme is responsible for coming in and phosphorylating and activating the myosin by taking a phosphate group from ATP, pulling it off, adding it to the myosin molecule, and then leaving ADP. This allows the myosin to become activated and now allows us to form the cross bridge with actin. And now we go through the cross bridge cycle of pulling our myosin molecules closer and closer together, and that causes the actual contraction of our smooth muscle cell. In order for us then to go through the process of relaxation, we essentially go in the reverse process. We have calcium detachment from the calmodulin. We are then able to pull calcium out of the sarcoplasm, and then we dephosphorylate the myosin by the myosin light chain phosphatase. And now we're back to a relaxated smooth muscle cell. Now, the one thing that we find with smooth muscle contraction is that the contractions are very slow and synchronized. And this is due to the gap junctions that are formed between the cells. And so if we remember back to earlier chapters, we talked about tight junctions and gap junctions and desmosomes. The gap junctions are bound together between cells by channels that allow a flow of ions from one cell to the next. And because of these gap junctions, it allows the cells really to contract as a plate or as a unit. we're going to find that some of the cells are self-excitatory, which means they depolarize without any external stimuli and essentially acting as pacemakers for these sheets of muscle. We also will see this in our cardiac muscle, where we have a pacemaker for your heart. The rate and intensity of contraction can also be modified by neural and hormonal control. Now, how is smooth muscle contraction similar to skeletal muscle? Well, one, it utilizes the sliding filament mechanism. It uses myosin and actin in order to cause a cross bridge, and we see the sliding filaments. We also see that the final trigger for muscle contraction before the cross bridges are formed is an increase in intracellular calcium levels. And then the other factor is that the sliding process is energized through the use of ATP. ATP goes through that dephosphorylation process on that myosin head in both cases. We also see that calcium is obtained from the sarcoplasmic reticulum via membrane channels from the extracellular space. So what are some of the functional categories of smooth muscle? Well, when we look at smooth muscle, we have two categories. We have a multi-unit and a single unit. With a multi-unit smooth muscle, we're going to see that they're located in large airways, large arteries, the erector pili muscles, as well as the iris of the eye. In multi-unit smooth muscle, we're going to see that gap junctions are actually rare. They're still arranged in motor units so that we are able to contract multiple muscles from a particular motor neuron. And then we also have graded contractions, which change the size of the contraction in response to neural stimuli. With a single unit or a visceral smooth muscle, this is what the common type of smooth muscle is. It's going to be stimulated to contract in unison as the cells are linked by gap junctions. So again, we're kind of seeing this contraction as a complete unit. Due to the pacemaker action and some of the self-excitatory components, we're often going to see some sort of spontaneous action potentials within them. The locations of our visceral smooth muscle or the single unit smooth muscle are going to include the walls of the digestive, urinary, and reproductive tracts, portions of the respiratory tract, and then most of our blood vessels. When we look at neural regulation of smooth muscle, we're really going to involve the neurotransmitter binding. And neurotransmitter binding aids in the increase in the calcium levels in the sarcoplasm. And this actually stimulates either a graded potential or an action potential. We'll talk about graded potentials a little bit more when we talk about the nervous system, but we can actually see whether the graded potential is actually going to be stimulatory or inhibitory. Again, the response then depends on the particular neurotransmitter released and the type of receptor molecules that fall on the muscle cell. So with skeletal muscle, the one thing we had was the neurotransmitter was acetylcholine. As acetylcholine crossed the synaptic cleft, it would bind to our ligand-gated sodium-potassium channels, and it would cause those channels to open, always causing an excitatory event for the skeletal muscle. In this case, the neurotransmitter can be multiple different types. And because of having multiple types of neurotransmitters, it can lead us to having multiple different types of effects. And so down here at the bottom, we will see that we have two examples. We have acetylcholine and we have norepinephrine. We know acetylcholine is excitatory in skeletal muscle, but what is it in smooth muscle? Well, when we look at acetylcholine in the example of a bronchiole, we get strong contraction or constriction of those bronchioles. So the space or the airway actually gets smaller and makes it harder to breathe. However, we also have norepinephrine. Now, norepinephrine has the opposite effect. It's inhibitory in the bronchioles, and so it actually causes dilation. However, norepinephrine also has a different function in most blood vessel walls, where it causes a contraction, and that causes a constriction of the blood vessels. So if we think about this, acetylcholine causes a strong bronchial contraction. Norepinephrine, or its very close cousin, epinephrine, has this inhibitory effect in the bronchioles, which causes dilation. So when we consider the EpiPen, we end up seeing that we use that in response to an allergic reaction. And what happens is in an allergic reaction, it causes that contraction or that constriction for breathing. By utilizing the EpiPen, that adds epinephrine to your system and causes this inhibitory effect in the bronchioles, allowing the bronchioles to dilate and allowing you to breathe easier. Here's going to be our multi-unit and single-unit smooth muscle. You can see with a multi-unit, again, we have the layers of cells with the synapses on top of each of the cells. In this case, with a single unit smooth muscle, we just have essentially the neuron with the vesicles going right through the middle part of this layer. And last type of muscle tissue we have is cardiac muscle. Now with cardiac muscle, these are going to be short branching fibers. They're going to have one or two nuclei in them. They are going to be striated, so they do contain sarcomeres. They have many mitochondria, and they do use aerobic respiration, which is why it's important for us to have a lot of oxygen. It is involuntary, so where skeletal muscle is voluntary, you have control over it. Smooth muscle is involuntary, you don't have control over it. Cardiac muscle is also involuntary, which means you don't have control of your heart. You don't have the ability to tell your heart to beat and then to stop and then to beat. It continues to move on as it chooses. We have intercalated discs, which help to join the ends of our neighboring fibers. And these discs contain desmosomes as well as gap junctions. Remember, desmosomes are kind of these spot welds, which allow for a little bit of movement, which is really important in cardiac muscle as the entire heart is beating. We want to have some sort of movement between the cells. But then they're also joined by gap junctions, which allows those ions to pass through from one cell to the next and allows the heart essentially to contract as one entire unit. The contractions are started by the heart's autorhythmic pacemaker cells, which have a self-excitatory component to them, but then it's also controlled by the nervous system. And so the heart rate and the contraction force is actually controlled by your autonomic nervous system. Again, the part of the nervous system that you do not have control over. And so this is what we see when we look at a cardiac muscle. Again, you can see the striations, you can see the components of a sarcomere. You can see that we have the sarcolemma around it, the desmosomes between the cell layers and the cells themselves, gap junctions to help join cells together. And then again, you can see these other components with the desmosomes, the intercalated discs, and then just the cell in general. And this will end up being the end of chapter 10. And so let's just make today's attendance code word simple, and it's going to be cardiac muscle. Okay, so the code words for today's attendance is cardiac muscle.